Good morning. I'm Bonnie Raindrop, and I'm Project Director for Pesticides in the Chesapeake Bay Watershed Project. This is a project of our organization, the Maryland Pesticide Education Network, and we want to welcome you to this last conference webinar session of PFAS and pesticides, exploring threats to people, food, water, and ecosystems. We're pleased that this year we have over 300 stakeholders registered for the conference, which shows that this topic concerns a growing number of people. Some of you have been part of this community and have attended these conferences year after year. Some of you are members of the project's working groups and help advance a variety of projects with us. We also have quite a few newcomers, and we're especially glad to welcome you to this community of stakeholders. All of us in some way have an interest in minimizing the impacts of pesticides. You all are experts in your own right, so the collaboration that takes place when we come together, even virtually, advances our understanding of the role pesticides play and more possibilities for how we can work together to more effectively limit pesticide impacts. So thank you for being here today. Next. Before we move on, please open your chat window, tell us who you are, and share something about yourself or a project that you're involved with that's related to pesticides. We all miss the networking we used to do when this was an in-person conference, but we'll do the best we can with chat. So please chime in with your thoughts today, and I'll send out the chat transcript for follow-up. And you'll also get the roster of attendees with their contact information to help you with further networking. We'll also use the Q&A module to pose questions to our presenters. So click on that and type questions in as we go. Whatever we can't get to live, we'll answer in text and send that along with the chat transcript next week. Next. Now I want to turn things over to our conference moderator, Dr. Greg Allen. We are so proud that Greg has been our conference moderator since the project's inception 18 years ago. Greg also co-leads our project's Research and Data Gap Working Group, which you'll hear more about. Greg is an environmental scientist at the U.S. Environmental Protection Agency, Chesapeake Bay Program Office. He's also chair of the Chesapeake Bay Program's Toxic Contaminants Work Group. For nearly a decade before joining EPA, Greg was in the field and at the analytical chemistry bench analyzing the occurrence of organic contaminants. Greg counts among his most meaningful recognitions a White House Closing the Circle Award, an EPA Gold Medal, and a handwritten thank you note from the Puget Sound Partnership for helping with their pollution reduction strategy. So welcome, Greg. Now I'll turn this over to you.
All right. Thank you so much, Bonnie. Welcome, everyone. Good morning to part 2 of our 18th annual Pesticides in the Chesapeake Watershed Project Conference. Hope you had a great Thanksgiving and that you're staying warm this morning, particularly if you're on the eastern side of the U.S. This is our second session. And this year we returned to this topic of the association among pesticides and PFAS, or per and polyfluoroalkyl substances. And again, today we have 3 fantastic speakers on this topic. I want to say thanks right away to Bonnie Raindrop and the rest of the team at the Maryland Pesticide Education Network for putting together again this year a fantastic program featuring some really fascinating presentations. So thank you so much to that for that. And just before we jump into the program today, we have a couple of things we'd like to do. As usual, the first thing is to revisit and reaffirm our commitment to the values that have sustained this project over time. So I'm going to pause for a moment so you can read these values. It's this commitment and our recognition of the social value that we all benefit from with the use of pesticides, while also recognizing that they present risk, and we want to come together to understand that risk and manage it. So we are sure that we will continue in that way and maintain our commitment to these values. The other thing that is special about this project and has helped to sustain it and make it so impactful is our work groups. And we have three work groups. The first is sustainable agriculture, next, research and data gaps, and the third is healthy alternatives. And as we typically do in session 2, we'll hear today from the leaders of these groups. They'll talk about work that they've completed over the last year since our last conference, and then talk about some future direction. And that direction has been informed by feedback that we've gotten from you all. And we'll show you how we're using that information. So we'll hear more about that later today. I want to point out here that we do have a vacancy for co-chair for the Healthy Alternatives work group. And you can see there what that work group's mission is. If that intrigues you and you'd be interested in becoming a member or leader of that group, please let us know. And Bonnie Raindrop would be the person to send a note to. All right. So one more thing we wanted to do before we dive into the program is to review some information that we got from you during our last session. So we asked a chat question. What were some of the ways that you were planning to incorporate sustainable practices in your holiday meals and holiday celebrations? Our first session was the week before Thanksgiving. So you can see here, we got a ton of great feedback in chat, and we did some affinity analysis on this. And we identified 5 themes that we were seeing: sustainable food practices, sustainable gifting, green kitchen and dining, home decorating, and community engagement in sustainability. So we're just going to pause here for a moment. You can review this list. Again, we were hoping that we would all be inspired by some of these ideas. Also, you can place into chat for us, maybe you had an additional idea about sustainable holiday practices. We'd love to hear any additional ideas, or if you see one of these ideas that you were able to follow through on, maybe you could share that example. And while you're doing that, reading the list and maybe adding a little more in chat, I'm going to point out that we saw a fairly strong theme here about organic fruits and vegetables. And we do have a web resource that speaks to that. And you'll see that here on the slide. Some of the work out of the Healthy Alternatives group is there. And otherwise, it's a resource that would, in particular, help us follow through on the use of organic fruits and vegetables for those of us that choose to do so. So we are just reading this list, maybe sharing a few more ideas or a holiday success that we had around sustainable practices. Thank you for everyone who's contributing to chat. Okay, yeah, we're seeing stories of using organic fruits and vegetables. In general, people liking these ideas, and we've got another major holiday coming up. So another opportunity to just be mindful of how we can minimize our impact. During the holiday season, it tends to be, well, kind of consumptive. So it's a great time to follow through and put in some effort to a more sustainable holiday season. Okay, well, we will appreciate anyone who has more stories to tell about their holidays or maybe something you're planning for the next holiday season. And thank you again for those who are back from session one and contributed to this inspirational list. Okay, well, let's go ahead now and dive into the program now that we're warmed up a little bit. So again, a great set of speakers today. We will hear from Dr. Vicki Blazer, some research around smallmouth bass and the presence of PFAS in those fish. Dr. Anna Marie Rule on some findings from a study on PFAS in human blood in the state of Maryland. And then finally from Dr. Alexis Temkin, information about the fate of fluorinated pesticides, their degradation, and what we know about the fate of those degradates of fluorinated pesticides. So a fantastic slate of speakers. And let's go ahead and start here with Dr. Vicki Blazer. Vicki received a BS in marine biology from Southampton College and a PhD in fisheries, aquaculture, and pathology at the University of Rhode Island. She completed a postdoc at the Veterinary College of the University of Georgia and then became the assistant unit leader and a faculty member of the School of Forest Resources at the University of Georgia. She moved to the USGS Eastern Ecological Science Center in 1992 as a research fishery biologist. She developed a program in fish health assessment in ecotoxicology and has had regional, national, and international projects. She's on the faculty and has directed graduate students' research at West Virginia University and Penn State University. She has authored over 200 publications and received awards from the American Fisheries Society, the Potomac River Keepers, and others for her research. We are always happy to welcome Dr. Vicki Blazer.
Good morning, everyone. So I'm going to present some of our data on land use associations with some of the PFAS we have been seeing in smallmouth bass in the Chesapeake Bay watershed and some nearby watersheds as well. Next. So since the early 2000s, we have been doing a lot of looking at smallmouth bass health issues as well as population declines in Pennsylvania, West Virginia, Maryland, and Virginia. We've seen fish kills, we've seen a high prevalence of lesions due to multiple viral, bacterial, and parasitic infections, which to us suggests that immunomodulation is occurring. We also see intersex, which indicates exposure to endocrine disruptors. And so starting in 2013, we conducted comprehensive fish health assessments in those states. And as part of that, fish were bled. We obtained the blood sample, and then back at the lab, we got plasma from those blood samples, and we aliquoted that and stored it at -80 in case we would have things down the road we wanted to look at. At 4 of those sites, we also did surface water samples, and they were analyzed for a large suite of contaminants, but that included pesticides, 105 different pesticides. They were sampled monthly most of the year and then bi-monthly in the spring from 2016 through 2019. Next. as PFAS became recognized as a potential issue and potentially contributing to some of the disease issues we were seeing in smallmouth bass, we took some of that archived plasma that we had collected from 4 sites in 2018. And early on, they were analyzed for 13 different PFAS. Now, as we move on, we're analyzing for more. But 4 PFAS were found in every single smallmouth bass plasma sample. And as you can see from the graphs below, PFOS had the highest concentrations. And if you look at the bar, PFOS is usually an order of magnitude higher than the other 3. But the other thing you see in those graphs is that there was quite a bit of site difference. So Antietam Creek, which is the black bar, had the highest of all of the different compounds. West Branch Macantango, which is in the Susquehanna drainage, had the second highest. And then the South Branch Potomac near Moorfield and Pine Creek, also in the Susquehanna drainage, had the lowest. Next. And when we looked at potential land use associated with those, what we saw was that, for instance, Antietam Creek had the highest developed land, which is often associated with exposure to PFAS, but it also has a high agricultural, the highest, 49%. West Branch Macantango, which was the second highest, had the second highest percent of agriculture. And then South Branch, Potomac, and Pine Creek have the lowest agriculture as well as the lowest developed land use. So that did make us think more about agricultural sources for some of these compounds. Next. And this is just a way to kind of look at it graphically. On the right-hand side, you see that graph again for the PFOS and the site differences. And if you look at both agricultural land use and developed land use, you see somewhat of the same pattern, with Antietam having the highest, West Branch Macantango the second, and then the South Branch Potomac as well as Pine the lowest. Next. So we did decide that all of this data did indicate that PFAS might be having an effect and that we should look at a broader spatial assessment. And so the map on the right gives you an idea of where the sites were that we collected. This includes the same 4 sites that I mentioned we did in 2018. We also had a site that's out of the Chesapeake, Cheat River in West Virginia. Then we added 2 extra sites in the Susquehanna. SS is the main stem Susquehanna near Selinsgrove, and CC is Chillisquaque Creek. And then the 2 highest on the graph, SC is Swatara Creek, and LNC is Little Neshaminy Creek. Next slide. And again, when we look at the upstream land use for these, Little Neshaminy Creek is actually close to Philadelphia. It's impacted by the Willow Grove military base, and so we expected it would have the highest levels, and indeed it did. It also has the highest level of developed land and a lower agricultural land use. On the right-hand side, EPA has put out a list of potential facilities that potentially release PFAS, and so we also looked at those. So if you look at the 2 that had the highest, Little Neshaminy and Swatara Creek, they do have the highest, and Antietam Creek also has a high number of potential point sources. But our interest was kind of in those middle ones, the West Branch Macantango, Chillisquaque, and including Antietam, where you have a high developed land use. And for some of those, particularly West Branch Macantango and Chillisquaque, you don't have many point sources, and developed land is also lower. So again, our interest was, what are the possible sources from agriculture? Next slide. This is just a spatial assessment and a graphical to show you again, the 4 PFAS that were found in every plasma sample. And what you see, the graph that I had previously was total PFAS. And what you see is that PFOS is often very similar to the total because it is always the highest level we see in smallmouth bass plasma. And again, even in this spatial distribution, PFOS is usually an order of magnitude higher than the other 3 compounds. And you can see there's a little bit of a difference from site to site for those other 3, but again, in general, or for all of them, Little Neshaminy has the highest level, Swatara Creek next, and then Antietam, and then the 3 sites that are more agricultural associated. Next slide. We also looked at those 4 sites that we did over time. We wanted to see, was there much change over time and from year to year at those sites? And as you can see, there is a little bit of change from year to year. Sometimes it's significant, sometimes it's not. But the same pattern among the sites stays the same. So Antietam Creek has the highest, irrespective of the year. West Branch, Macantango, the next highest, and then South Branch and Pine tend to be similar. Next slide. So what are some of the agricultural sources that we're looking at? Obviously, this conference, we're really focusing on pesticides and PFAS. But I do just want to point out that we are very concerned about biosolids as well. And we do think that that is an important source in lands that are primarily agricultural. And as far as pesticides, if you were here at the last session, you heard a lot about the sources within the pesticides. So there are pesticides that have PFAS as active ingredients, also some that have PFAS as inert ingredients, and then there can also be leachate from containers. So a number of different sources in pesticides. Next slide. So according to a recent paper, there are 66 registered PFAS pesticides where PFAS is an active ingredient. And as I mentioned before, we had data in surface water for 105 different pesticides that we had analyzed at our 4 temporal sites. And when I looked through this list, at least 16 of those were considered PFAS pesticides. And so I looked at that data to see, were we finding those at our sites? Most of those 16 were not detected, but 2 were detected frequently at the 2 sites, Antietam and West Branch Macantango, which had the highest PFAS concentrations. And those were fipronil and its metabolites, and that's an insecticide, and then norflurazone, which is an herbicide. Next slide. Fipronil was actually detected in all years throughout the year at Antietam Creek, except for a couple non-detects in early spring in some of the years. It was detected only once in 2018 at West Branch Macantango and also at the South Branch Potomac, although at lower levels, and there were no detects at Pine Creek. The norflurazone was detected in both Antietam Creek and West Branch Macantango, and no detects were found in the South Branch or Pine Creek. Next slide. But there were also some other things I think we need to think about as far as this. There is one pesticide, it's an insecticide, sulfuramide, that's used in a range of products to control ants, roaches, and termites. Its use in the US is actually restricted, but this is one of those that is a good indicator of how metabolism of some of these substances, for instance, the ETFOSA, is metabolized to PFOSA, which then is metabolized to the PFOS. And it has been shown to be a major source of PFOS in biosolids. So even though we didn't detect it in water, and actually it was one we didn't look for, it's an example of things that we need to be thinking about, because obviously we're not looking at every single pesticide that we could look at. The other thing is that we did not find bifenthrin, which is one of the PFAS pesticides, in water, but it was detected in sediment samples, and it was actually one of the few that was detected in young of the year smallmouth bass tissue. So again, we need to think of sources. Next slide, please. And when we think of sources for a fish like smallmouth bass, we have to consider not only the surface water, but sediment and also diet or food web issues. So smallmouth bass, particularly the males, build and guard nests during the spawning season, so they're in association with the sediment, and then young of the year are also associated with those bottom sediments. So any of these compounds that are actually ending up more commonly in the sediment than in the water or in the food web can be major contributors as well. Next slide. So in conclusion, we are finding PFAS in smallmouth bass, even at sites that are primarily agriculture and forested land use. We do have evidence that pesticides and potentially biosolids are contributing to these levels, and they could come from both surface water and sediments. And the other thing, that I think needs a lot more work is we find PFOS at significantly higher concentrations than the other PFAS. But it is one of what's called a terminal PFAS. And so many of the other PFAS can be metabolized to the PFOS. And those are things we definitely need to be looking more at. Next slide. And so I'd just like to end by saying that this was funded by USGS Ecosystems Mission Area, Environmental Health and Land Management Programs. We are continuing to look, so all the fish that we have PFAS data for in the plasma, we also have lots of biological endpoints from the cellular to the molecular to the organism level. And Heather Walsh and Cheyenne Smith, who work in the lab, are taking that data, and we're trying very hard to see, are we seeing effects from any of these levels that we find in smallmouth bass? And then lastly, I'd like to thank the many state biologists in all the states that work so closely with us and really allow this work to be done. Next slide. And if there's any questions, I will certainly take them. Thank you.
Thank you so much, Vicki. Amazing work that you do out in the field. It's always fascinating to hear about it. So we'll have a few minutes for questions and conversation here. And I'm going to follow with one of your last points here, because one of our attendees had asked about effects. And I understand you're just really working on that. Do you have any guesses, any insights right now on effects that you could talk about, or do we need to wait for that work to be done?
Well, actually, some of the interesting thing, Cheyenne Smith has been doing a lot of work with immune function assays, and we have seen correlations. And of course, when you're looking at wild fish, it's all we have right now is correlations. But she's taking some of the concentrations that we're actually measuring in the plasma into the lab and looking at those concentrations on cells that we take out of smallmouth bass that we've kept in the lab. So they should be fairly non-contaminated. So we do feel that we are certainly seeing effects on the immune system. So that's the one thing we've really been able to see. We see a lot of changes with gene expression, but we're still trying to tease out what does that mean.
Okay, well, we'll look forward to more findings there. So a couple more questions that came in. Do we have any data on any other species of fish in the watershed? Are you aware of any other plasma for other species other than smallmouth?
I am not aware of other plasma in the Chesapeake. There is work, some down in North Carolina, some other areas that have looked at other species like striped bass that we do certainly have in the Chesapeake. Um, and one of the things that's a little difficult to compare, a lot of the state agencies are looking at fish for human consumption. So they're looking at the concentrations that are in fillets or in mussel. And one of the things we have found looking at different tissues is that plasma and blood does tend to have the highest levels, liver next, and then we've also looked at gonad and some other tissues. Muscle tends to have the lowest, which is good news for people that are eating the fish, maybe not such good news for the fish.
Okay. And so there's a question here about how smallmouth are becoming contaminated. What are your thoughts? Well, you've talked about this a bit. It's a combination of being close to sediment and also their food sources. Is that how it's happening for them?
Yeah, I think, all of them could contribute. Smallmouth bass are one of the top predators. So obviously I think food web for those types of organisms plays a big role.
Hmm. Okay. Yes. And folks asking about people consuming these fish. And my understanding is that smallmouth is not as much a fish that's sought out to be consumed, but it's a very important fish economically because there are guides and a lot of people that love to fish for them. So economically, their health is really important.
Correct.
Okay. Um, just one last question here, Vicki. Uh, Could we develop a model that would help us take plasma concentrations and predict the level that would be in fish tissue in particular?
Well, it's kind of interesting because we were working with Pennsylvania DEP for the Swatara Creek and the Little Neshaminy, and that's where both of those sites, we collected different tissues to see could we come up with that. And it was kind of interesting because at Swatara Creek, the highest concentration was in the plasma, the next highest was in whole blood, then liver, then gonad, then muscle. At Little Neshaminy Creek, it was different. We had higher concentrations in liver and whole blood than we did in plasma. And I haven't yet come up with an explanation for that. um, but it might be possible to do the conversion to muscle because muscle was always the lowest at both sites. But
Hmm.
when I ran just simple statistics to see could I come up with any kind of, you know, conversion, it didn't really work. But those are
Okay.
just two sites. So we may need more data.
Hmm. Okay. Well, hope you'll keep us up on that as well. Uh, so we do have another couple minutes here. And Vicki, I noticed a major point that you make here is that conversion is important. Conversion may be what's, for example, contributing to high PFAS eventually in the tissue. Not a whole lot we can do about conversion once the pollutant is in the system. But what can we do to keep it out of the system? Are BMPs that we're using for things like nitrogen controls, things like forest buffers, no-till agriculture, those kinds of BMPs, should they be helpful in keeping PFAS out of surface water?
Well, we did look at a few of those and we didn't see a big effect. And of course, again, we only had those 4 sites where we had some long-term data to be able to look at some of those things. Um, I think it gets back probably more to the sources, you know, and again, focusing on agricultural sources, because certainly we're seeing PFAS, and I would say the agricultural sites are kind of the moderate, the intermediate. You know, we have the very high at things like Willow Grove, obviously, and then much lower at some of the more forested sites. So ag is kind of in the middle, and I do think we have to look at things like biosolids and pesticides as far as sources in agricultural sites.
Yeah. Okay. Well, certainly working the root sources is the most effective strategy. And in the meantime, hopefully there are best management practices. Thank you, Anna, for that question in chat. We tend to get a little loose with the acronyms here. So thank you. Yes, those are best management practices, the things that folks can do on the landscape that hopefully reduce the amount of pollutant that's coming into the system and becoming bioavailable in the case of the toxic contaminants. So all kinds of agriculture and developed land actions that reduce pollutants we refer to as best management practices. Okay, well, thank you so much, Vicki, as always, and look forward to some updates on this work in the future. And we'll be hearing from you a little later for the Data Gaps and Research Work Group. So look forward to that too.
Okay.
Vicki, there's also some questions in Q&A and in chat that if you have a minute to respond to, we would appreciate that. All right, well, let's roll right into our next presentation here. I am introducing Dr. Anna Maria Rule. She is an assistant professor at Johns Hopkins Bloomberg School of Public Health, where she obtained her master's in occupational health in 1996 and her PhD in environmental health sciences in 2005. Dr. Rule is an expert in exposure assessment of airborne, environmental, and occupational hazards, including in agricultural, urban, and nosocomial environments. She has led projects to evaluate exposures to biological aerosols, electronic cigarettes, as well as indoor and outdoor air pollutants. Dr. Rule's main research goal is the development and evaluation of novel sampling and analysis strategies for the assessment of exposure to air pollutants. She's currently director of the Environmental Exposure Assessment Labs, where she develops and applies methods for the assessment of exposures to adult and pediatric populations. She has experience working in multidisciplinary projects that involve collaboration with researchers from other disciplines and is passionate about addressing the root causes of environmental justice. So we welcome Dr. Anna Rule.
Thank you so much, Greg. So I am going to talk to you about some preliminary results. I know there's a lot of people here that are interested in this, of a project that we did to assess prevalence of per and polyfluoroalkyl substances, or PFAS, within the blood of individuals throughout Maryland. This is a project in combination between the Maryland Pesticide Education Network and Johns Hopkins University, with very generous funding from the Jacob and Hilda Blaustein Foundation. Next. So I want to start with my acknowledgements up front. First, I really do want to acknowledge this would not have been possible without the research team that did a fantastic job. I was just, sort of like I'm going to present some of the results, but they did the heavy lifting. So Caitlin Series is a project co-principal investigator. She's assistant professor in the Department of Health Sciences at Towson University. Mr. Sean Lynch is the project director and integrated pest management in healthcare facility project for MPM. Dr. Diana Egnor, she did most of the data analysis for this study and plotted all of the figures that I'm going to show that have graphs. She plotted them. Mr. Jack Solomon is a GIS expert, and he did all of the maps that I am going to present. And doctors Michael Ichniowski and Vicki Blazer are project advisors, and this would not have happened without their vast expertise on PFAS, and both in human and the environment. Next. So briefly, I'm going to tell you what PFAS are, sources, what we know about levels in the blood in the US population, and some of the health risks that we know. The goals of our study, the methods that we used, and I'm going to try to get as soon as possible to the results, and a brief discussion and next steps. Next. So as we have been hearing, PFAS are known as forever chemicals because they really resist degradation and breakdown in the environment and in our bodies. They are found in many of the consumer and industrial products that we use, including, among others, very, very common is nonstick cookware, water and stain repellent fabrics, and some foam for firefighting, but interesting for this group of people is some pesticides, as Dr. Blazer just explained. Next. So since 1999, the CDC, through the National Health and Nutrition Examination Survey that you may have heard happens every 2 years, has been measuring PFAS in the blood of the US population. So we have here a graph of the last, since they have started measuring PFAS since 1999, all the way to 2018. One important point to make is that about 97% of the US population that have been tested, which is representative of everybody, have PFAS in their blood. Due to some health concerns with a few PFAS and PFOS and PFOA in particular, they have stopped manufacturing them and substituting with other PFAS, which is how we can attribute this decline in some of the PFAS. But I just want to caution that this is a national average that hides a very wide variability. This is not representative of everybody everywhere. And they only tested a few selected PFAS. So in our study, we expanded to test a few more and to focus in Maryland. Next. So even though we're still studying PFAS and the health effects, and we don't have a full understanding of what exactly they do to our bodies, there is sufficient evidence of an increased risk of cancer, especially kidney and testicular, weakened immune responses, elevated cholesterol, liver damage, and decreased birth weight. And this is especially important. We have to keep in mind always the vulnerable populations like the pregnant women, children, immunocompromised people, and people in environmental justice communities, which are impacted by many other sources. Next. So the National Academies of Sciences and Engineering and Medicine in the last couple of years came up with a group and made recommendations for clinicians on what to do. We are beginning to be able to test these PFAS in the blood of people, but we don't know what they mean. So based on the sum of PFAS, which we're going to call the NASM, or NASM PFAS, in the blood, they came up with these 3 tiers. So if you have less than 2 nanograms per milliliter in the blood, your physician is not going to recommend anything different than what's typically done. Between 2 and 20 nanograms per milliliter, the clinician is going to encourage exposure reduction, see where you can find some sources that you can reduce, and screening for high blood pressure, hypertension, and breast cancer. And above 20 nanograms per milliliter or higher, then some additional tests would be required, including some thyroid function testing, assessment of kidney cancer, testicular cancer, and colitis. Next. So the goals of this study, as I mentioned briefly, are to characterize the concentrations of 45 PFAS chemicals in the blood of a sample of residents in Maryland. The second goal is to evaluate factors that potentially contribute to these exposures. So we asked participants about proximity to PFAS contaminated drinking water, some lifestyle recreational exposures, dietary preferences, and occupational exposures. And then we computed summary statistics for all of these to show you some of the data. Our hypothesis was that certain populations are disproportionately exposed to PFAS pollution across the state of Maryland. Next. So we recruited 42 participants from across Maryland. It was a convenient sample with a goal to recruit at least 5 per region. Participants answered a questionnaire through Qualtrics, so that we can get information about their age, gender, race, where they live, and some behaviors that expose to PFAS. Then Eurofins, working with Eurofins, which is a commercial lab that tests for PFAS, they sent each participant a kit to self-administer a finger prick test. Participants sent their blood sample back to the Eurofins, and they measured the 45 PFAS. Each participant received the results back within 2 weeks, and then we are receiving all of the results so that we can compute make the statistics. And then our team analyzed all data to assess associations between the PFAS in blood and socioeconomic and behavioral factors. So like using SAS statistical package to plot the PFAS and ArcGIS to map the geographic distributions. Next. So here go the preliminary results. Next. These are the first 32 participants. We don't have the whole picture yet, but it's most of them. So the first way in which we thought to express this data, and there's many ways to look at it, but this is called a cumulative percent distribution function, which shows you each of these points is the value that was reported for each of for participants for the total PFAS, which is the value that is used for these breakdowns of thresholds in the blood. So we can see 2 of them, 2 participants had less than 2, which is that first vertical line, and then 20 micrograms per nanograms per milliliter is that other vertical line, and 3 participants are higher than that, had levels of PFAS higher than that in their blood. And the rest of the participants, 27 of them, fall between these 2 and 20. And so there's a huge range, as you can see. One thing that is interesting about this graph is that you can tell, for example, what is the median. If you look at that 50% in the middle, if you go down to the horizontal axis, you can see that 8 nanograms per milliliter was around where the half of the population of our sample is located. Next. So another way to look at that data is the same 32 participants is with these box and whisker plots that show you the distribution of the blood concentrations detected. And in this case, we plotted by gender. So we have on the horizontal, we have the different PFAS that had at least one person test positive for them. And on the vertical, we have the blood serum levels in nanograms per milliliter. So by PFAS, we have these pairs of blue and red. The blue are the males, the red and the females. And I have added these red horizontal lines on the first 2, which is for the ones that we have national averages. So the national average for PFOA, which is the first pair, is 1.4 nanograms per milliliter. And as you can visually see, there's really no difference between the middle of the boxes. The boxes are very overlapping. The only difference is that the females have slightly more wide distribution, but that doesn't mean anything really. But for the second one, for the total PFOS, You can see that most of the males fall above the average, the national average, and most of the females fall below. As Dr. Placer mentioned already, the total PFAS, which is this NASEM third pair, follow very closely the total PFOS or PFOS. The rest of the boxes we put here just for information, but there's no difference between males and females, and they are lower in general than the PFOA and PFOS. Next. Ooh, okay, the colors are completely different here than what I was expecting. But here we have another way to look at that data. We have the age of the participants on the horizontal line. So our youngest participant was 29 years old, and our oldest participants were 75. In light gray, I hope you can see, are the males in our study, and in blue we have the females. The females actually, for this total PFOA, is the only one that had a correlation or association between age and concentration. So the older the females in our study, the higher they tended to have concentrations. But there was no correlation for males, and that correlation did not exist for any of the other PFAS that we looked at. Next. So then we plotted the PFAS serum concentrations by region of the participants where they lived. So we have here in the first set of boxes, we have total PFOA. In the second set, we have the PFOS, and then the NASEM total in the third blob set. And as we can see here, the only one that was slightly higher are the people in the southern region of Maryland. So just keep in mind, if you see these numbers in the middle of the graph, we have 10 people in the blue box, 13 people in the central region of Maryland, which is the red, 4 people in the eastern green, only 3 people in the southern region, and 2 people in the west. So we were trying to get a few more people from these regions to increase our numbers. Next. So look at that same information on a map. We see that the PFAS in serum by region and age ranges. The darker color is the age, and the bigger the dot is the higher concentration. So there's no really association. You can see where we have the participants, but we didn't see a big association between age and region. I mean, concentration and age, just the southern region. Next. Then PFAS by income ranges is the other way that we looked at it, at the data. And again, hopefully you can see just visually, there's no association. We did not see anything. There's no association. Another thing to keep in mind is we have only a few in the lower income ranges, so we also cannot say a lot more about this. But we have 7 people that in our sample that earn less than $75,000. Then in red, we have between $75,000 and $150,000, and in green, greater than $150,000, which is only 11 people. Next. And this is the same thing plotted in a map. So here, Jack plotted here the social vulnerability, which is an indication of how vulnerable you are to environmental impacts, social and environmental impacts. And in circles, in purple circles, we have the higher concentration, the darker the circle. And we really don't see any association. You don't see the darker circles where you have the darker areas. So by census tract. So no real association there. Next. So in conclusion, every one of the participants that we recruited had PFOS in their blood. The males in our study have slightly higher PFOS concentrations than females, and there were no difference really in the PFOAs. The age of females in our study has a weak positive correlation with higher PFOA concentrations. So the older people tend to have higher concentrations, but there were no other correlations with other PFAS. We also found the highest concentrations of PFOS in our participants from the southern region. And there were no difference really in PFAS concentrations either by income or by water supply, which we also plotted, and there was no correlation. So definitely more research is needed to confirm the differences that we see in this population. It's a small sample size. So next slide, we have a few of the strengths and limitations of our study. Some of the limitations are really that it's only 32 participants of the whole state of Maryland. So it's a very small sample size. It's not necessarily representative of the whole state. We don't have equal representation of all the regions, or equal representation of the ages, or equal representation across the income brackets. So we can see some trends, but we really cannot say anything else about that. But some of the strengths of our study is that it's one of the first studies to report 45 PFAS. In Maryland, some of the PFAS in pesticides cannot be measured in the blood, but it's a snapshot of Maryland areas of concern. So hopefully we can follow up on this. Thank you. Next slide. I will be happy to take any questions.
Okay, Anna, thank you very much for the update on this study. And yes, we have about 10 minutes for some Q&A and discussion here. So let's start with some of what is in the Q&A set here. So you showed that national average graph that showed particular PFOS coming down over time. And I think some of us are wondering if that coincides with the time when, to my understanding, it was a voluntary stoppage of using PFOS and OA in products and in manufacturing. The legal stoppage, I think, just happened recently by EPA, but it was voluntarily stopped. Do those dates coincide with the drop that we see?
Yes, and I apologize. I kind of like skip a little bit over that slide, but yes. So the PFOS voluntary stop was in 2002, and the PFOA stopped in 2015. So the declining trend does coincide when they stopped, but that is, remember, only in the U.S. and they may still be present in other products, but we do see that decline, which coincides with the stop. So it works.
It works.
That it works, yeah.
Yeah. Certainly does look like it demonstrates that. And now we have to be mindful, though, of other PFAS that can metabolize into PFOS, as we heard from Vicki and others. And so that may continue to contribute to PFOS in blood, but it looks like that voluntary stoppage really had an impact. We'll see if there is more of that. I know some states have moved very aggressively to ban use of other PFAS in products. So, okay, a couple other questions here. Asking about the income question. How did you quantify income in the study?
It was a self-report. So we asked people to, and it wasn't like an open question. We had pre-specified ranges, do you make between this and this and this and this? And so people selected the one that they belong to.
Okay, so that was self-reported. Okay, great. And how about future studies? I guess we're all interested in where this could go, and recognizing, as you have, that it's a limited data set, and you already said we need more data. So Any plans? What are the plans at this point to expand or maybe go to other states? And we have a participant from Delaware who's asking, would be interested in collecting data in Delaware. What are the next steps here?
So, as many of you might be aware, this mostly is driven by grants. So either you need money from a foundation or you need money from the government. And so we have already applied. We have a couple of grants in consideration, one by EPA and another an internal grant at Hopkins to specifically look at PFAS in Maryland. So we are waiting for the results. So we will keep doing that. And of course, this data that I showed it will be really helpful as preliminary data to show feasibility and to show the variability, so that there's more interest in either the government or foundations to fund future projects.
Okay. Yes. Well, I hope that happens, particularly including that social vulnerability piece of this and further investigating whether this is an environmental justice issue or to the extent that it is. That I think is really novel work there. A couple of questions here. Now, we know we have a limited data set, but you were showing that PFAS increases by age in women. That may be true for men also, but is that bioaccumulation happening right in front of our eyes?
Right. This was our hypothesis for everybody, not just for women. We were kind of like surprised that we didn't see the same result for men, and it could be this something in the specific participants that are participating in our study, but it is true that women in general, we tend to have more fatty tissue, and PFAS are lipid soluble. So maybe there's something going on there that we tend to accumulate more. But I leave that to the physicians and the biologists in the group. But the surprise was not that we found that correlation in the women, but that we didn't find it in the others. So I think we need just more data so that we can see why that might be.
Yeah. Wow. Okay, that'll be interesting to pursue for sure. One last question here. This has to do with TFA, trifluoroacetic acid, and it's a contaminant that is getting a lot of attention now because it looks like it's present in drinking water and other places, and we didn't really have a great understanding of its presence. But is that in your analyte list?
No, I don't think it is. No.
Yeah.
So
Right.
part of the problem with these new, which is what's interesting, I think, of our study, but we are learning as we go. There's more technical capabilities, better detection strategies, better methods for testing some of these PFAS, and unfortunately, the way it works is unfortunately manufacturers are able to release them into the environment and sell products with them without a lot of testing. So there's some minimal testing, but not toxicity testing. So we're playing catch up a little bit, but we did not have TFA in our panel.
Okay. Well, we got a lot of people here thanking you for this work and for this update. There are a few other questions that we didn't get to. So if you could, Anna, visit the Q&A and the chat, you might be able to offer a little bit more. Lots of applauses and a lot of love coming out to you for this work. So thank you so much for that. And Dr. Rule's contact information is also in chat now. And maybe we can keep that conversation going there in chat and Q&A. Appreciate you doing that, Anna, and thank you very much for the update. We look forward to bringing this back for future conferences and see where this work leads and see how it compares to, I'm sure, work that's going to be done nationally that will add to a large data set about the presence of these pollutants in humans, tissue,
Yes.
tissue and blood. So maybe over time we'll see these things come together and you will be contributing to a larger data set. All right. Well, thank you again, Anna. All right, here's our next element in our conference here, and we're going to do this while we take a break. But before we suggest that you take a break, I want to remind you that we asked this question also in our first session, and it was, what would you like to learn more about with regard to PFAS in pesticides? We did some affinity work on this, and you see some categories: PFAS presence and testing, mitigation and health solutions, health effects of PFAS, PFAS and pesticides, advocacy, public engagement, and then research and policy development. So what we're going to do is take our break for 5 minutes. We're going to run some information during the break about folks that help get this conference underway and get it done. And then when we come back from the break, we'll look at this list for a few more minutes. And in particular, we're going to be considering which of these needs or interests line up well with our work groups. And when we get this information from you all, we really try to put it to use. And in this case, this will inform the work of our work groups going forward. We'll be looking through this list and trying to see some priority items that would lead to new work for our work groups. So when we come back, we'll mark this up a little bit with regard to our work groups, talk about it, and then we'll hear our third presentation today. So let's go ahead and take a 5-minute break. Welcome back, everybody. We're going to go ahead and get started. So we asked this question in our last session, what would you like to learn more about? And we got a phenomenal response. You can see that this really is an impressive list of needs and interests among the community. What we're doing here is marking up these ideas with a few that really speak directly to the work of our work groups. We have agriculture, data gaps in research, and healthy alternatives. And some of these really speak directly to those topics. There's plenty of science in here with regard to testing, trying to understand fate and cycling. There are some healthy alternatives and how do we minimize the amount of PFAS that's coming into the system, ending up in some cases in our fish and compromising the value of that resource. Some suggestions around education, and that's certainly the kind of work that this project set out to do, awareness building and education around presence of pesticides and trying to come together to understand the risk that they present and how we can minimize the risk. There is more here on human health impacts and ways to mitigate and reduce the levels in humans once we've determined that we're in those concentrations that NASEM tells us are of concern and should be managed. We don't really do human health as much in this project, but we certainly do education and awareness building for healthy farms, healthy communities, healthy watershed. So that certainly does fit. And a few of these ideas speak to research that we feature at this conference, and we will continue to feature. So these ideas will help us with planning next year's conference and future conferences, particularly presuming that we keep some PFAS, at least PFAS updates in our conferences, if not a major theme. All right. Well, we want to thank you for this input, and we are showing here a couple of examples of how we're going to pass this on to our work groups, and they will consider it for future work. We would absolutely love to have more members in our work groups. There is an association between the number of people and the amount of outputs that we create. The work groups have generated products and outputs that have been really impressive and useful. And we get more of those when we have more people that can work on them. So really would love to encourage everyone to think about joining us in this work. It's not a lot of meetings. It's not a very heavy load, but fascinating topics. And we always welcome new participants. And remember, our Healthy Alternatives Work Group has a co-chair vacancy. All right. Well, again, thanks for this input. We're going to use it. I hope our regular and ongoing participants can see that when we ask you for information, we really try to put it to use. And you're going to see even more of evidence of that in our next piece, which we will go ahead and get to. And I said we had only one more presentation, and that was a mistake. We have our work group updates and then another presentation. Okay, so this is our time when we have our work groups report out on their progress and some future direction. So we're going to go right around on this, and we're going to start with healthy alternatives. And Veronica Corella, our chair there, is going to give us an update.
Well, thank you very much. Good morning. My name is Veronica Corella. I'm the Legislative Director for the Maryland Children's Environmental Health Coalition, and I'm a co-facilitator of the Healthier Alternatives Working Group. And I'd like to take a moment to thank Steve McDaniel for his many years of service as the work group's co-facilitator. And we are currently seeking a new co-facilitator, so please contact Bonnie or me if you're interested in joining the team. So during the past year, the Healthy Alternatives Working Group continued to focus on a number of ongoing projects, as well as to begin work on some new opportunities to explore healthier alternatives. One of our new projects has been our review of Maryland's 1997 Integrated Pest Management, or IPM in Schools law, which prioritizes non-chemical means of pest control only to be used as a last resort, thereby providing healthier alternatives to chemical means of pest control in schools. We are contributing to this effort to ensure that children are fully protected under the law by reviewing current policies, plans, and practices across Maryland's 24 public school districts. We are collaborating with the current administration and our MDA secretary and his staff in this effort, which is a really positive and great development for us. We made some progress by working on with both MDA and the Maryland Children's Environmental Health and Protection Advisory Council, which is known as CPAC. After meeting upon request from MDA, we're working to provide MDA with a model policy and model plan to create a state template for all counties to ensure the law is properly being implemented. And we collaborated with CPAC's Schools Working Group to analyze the existing county plans. Some of them were almost 27 years old for each component of the law, and found a lack of consistency in county schools in abiding to all of the components of the law. So we're encouraging MDA to collect the records of pesticide applications for any school so that the public and others, like our working group, can be aware of what pesticide applications are made at schools in real time in order to assist healthcare providers in addressing children and staff who may have been impacted by exposure, but also to allow monitoring and review of the pesticides used, because that enables analysis for the reason that the pesticides were used, and it opens up the path to researching the healthier alternatives. Then we took a fresh look at our pesticides, the risk prevention and healthier choices booklet, which was originally introduced and widely distributed in printed form. We developed a new social media toolkit campaign based on this booklet and updated MPEN's website section that provides alternatives to pesticides. The toolkit is intended to make it easier to post specific information from our guide to healthier choices, such as information on risks, reducing exposures, alternatives, and how to make your yard safer, etc. We also did outreach to Maryland schools for our Safer Disinfectants campaign, because disinfectants are actually EPA registered pesticides. Our team sent letters to school district employees about our Safer Disinfectants resource to help districts make safer selections versus increasing risk disinfectants in schools where children and staff may be exposed. We continued our annual work on our Managing Mosquitoes Without Pesticides campaign. Posting using our toolkit are shared widely across platforms, and we do encourage you to please share the posts on your own social media platforms. Next. Our working group continues to make progress on the Jamboard ideas from our prior annual meeting, including educating the public on pesticide dangers to humans or public health and wildlife and pollinator habitats, as well as educating the public on safer alternatives to pesticides and tools such as MDA's Pesticide Sensitive Individual Registration Program, which has been available since 1988. This right to know program allows for prior notification when a pesticide hazardous to your health is applied by a commercial applicator to a property adjacent to your residence. We continue to develop brochures and flyers to put in public libraries and distribute in other ways. And we're working to educate the public on the connections such as healthy soil carbon capture and how pesticides destroy soil health. And looking forward to 2025, we plan to complete the IPM in schools model policy and plan and support MDA in adopting and implementing these updated versions to ensure children continue to be fully supported under the IPM in schools law. We will continue to publish and share our social media toolkits, and we will develop new social media toolkits for turf flags, that little yellow flag you see there posted each time a hazardous pesticide is applied to turf. And also for organic lawn care, which doesn't require the posting of the little flag. We also plan to develop outreach information on the new pollinator habitat law, as well as updated outreach on the IPM in schools law. Next. So finally, ways you can get involved. You can certainly post and share our social media toolkit. You can share ideas you have for opportunities to reduce risk and educate the public. And you don't have to join our work group to do that. You can just pop us an email if you have any suggestions you'd like us to look into. You can join the project's Healthy Alternatives Working Group for 2025, and you can consider being a co-facilitator. So if you are interested, please reach out to Sean or Bonnie or me, and we would be happy to incorporate your ideas or suggestions or you into our work group. Thank you.
Thank you, Veronica. Fantastic work. Really appreciate that update. Veronica's contact information is in chat. Thank you very much for that. All right, let's move to our next work group, Sustainable Agriculture.
Good morning, everybody. Great presentation so far. Really appreciate all that great information. I'm Cleo Braver, and I'm the owner with my husband of Cottingham Farm, which is an organic farm in Talbot County on the Eastern Shore. And I'm facilitating the Sustainable Ag Working Group along with Errol Maddox. This year, we've been working on a couple of projects where we're in the process of developing a landowner action plan document that recommends ways that stakeholders, including farmers, but not limited to farmers, those who rent farmland, even residential landowners, can improve the health of the land and positively, rather than negatively, contributing to the Chesapeake Bay watershed at the same time. You know, we've been grappling with the challenge this year of connecting with these communities. And if you're one of these stakeholders or work with them, please reach out to us. We'd love to talk. It's important to note that, you know, residential land is a very close second behind farmland. So we can all be part of the solution. One of our working group members, Steve Kraszewski, is developing a video series on organic regenerative farming practices, which is aimed at farmers who may be interested in transitioning from conventional to organic healthy soils practices. He's completed the first video on strip-till corn. It's in the stages of final production, and we expect a number of additional videos as well. And lastly, we're finalizing a social media toolkit for educating the public on organic regenerative farming practices and growing practices for those of you who don't have farms but do have a little bit of land. And the benefits of these practices to the environment and our health. It's designed to, you know, help us drive demand through educating consumers for why they should seek out and support farmers who are using these practices and foods that are grown using these practices, because we do all eat, so we can all have a huge impact even if we don't own farmland, because we are wielding our forks. Next slide. Yeah, thank you. The working group is still using the Jamboard ideas that were generated by our conference discussion as a guide to our work. The ones you see here on this list are the ones that are the dominant themes, and our conversations continue about how to best move forward. Our landowner action plan is sort of being broadened to a land stewardship action plan, and we're going to be distributing this to relevant stakeholders in Maryland, i.e. all of us. We also plan to develop and create more short clips on organic regenerative farming practices. It's kind of a how-to to help farmers and landowners or land renters who are considering these practices on their own land. And we'll launch our regenerative organic farming social media campaign to help spread the word about this. So watch for an email probably in January with a link to the toolkit and a request that you share the campaign in your own social media pages and reach out to your community of folks. Next slide, please. The Sustainable Ag Working Group is, again, as I said, for farmers, it's for landowners, it's for renters, it's for people who eat, and it's for all stakeholders in the agriculture ecosystem who are interested in educating landowners about these key issues and concepts and how they can be a part of transformative change. It's a great place to share your expertise on farming practices and growing practices and even legal issues for farmers. So please look for an email that's going to invite you to join us at our next virtual meeting, which will be in the first quarter of 2024. Thank you.
Right. Cleo, thank you so much for that update. That meeting will be first quarter of 2025.
Yes, 2025, yes.
Okay, great. All right. Thank you so much for that. Cleo's contact info is now in chat. And there are some requests for links to toolkits. So anywhere that we have links to some of what these work groups have produced, we will try to share those. So maybe we'll get some of that into chat here shortly. Thank you very much, Cleo, for that report on the work of this work group. We have one more work group to hear from. That's the Research and Data Gaps work group. And we're going to hear from Sean Lynch on this.
Thank you, Greg. Good morning, everyone. I'm Sean Lynch, and I'm the IPM and Healthcare Facilities Project Director at Maryland Pesticide Education Network. So I'm one of the coordinators in the work for the work groups, and in the Research and Data Gaps working group, I support our co-leads, which one of is Greg Allen, and the other is Vicki Blazer, and both of whom we've been honored to see present at the conference this year. So in the past year, the Research and Data Gaps working group met several times to discuss and finalize this report, communicating recent pesticide research findings to inform stakeholders. And the working group has been focused on this for the recent 2 years. And as you can see here, it summarizes relevant studies relating to the Bay watershed and pesticide use. So the report also includes important potential actions for the Bay Cabinet and other stakeholders that they can consider in their efforts to protect the Bay watershed and those of us that live within it. Next slide, please. So in the last year, we also looked at the Jamboard ideas as well, which were generated by the conference discussions to help inform the work for 2025. And the ideas shown here, the group agreed were a priority, with focusing on updating the research report each year moving forward as well. The discussion about the report this year also focused on to whom it will be distributed and how. And so looking forward to 2025, we plan to put out a press release, distribute the report to the Bay Cabinet and to all relevant stakeholders. And in addition, the group plans to develop a presentation as an output and work with stakeholder organizations to disseminate this information. The group also plans to publish the matrices, which are compounds of concern that were an outflow of the report as well. And then continue to do that development of the annual updates for the research report going forward. Next slide, please. So what ways can you become involved in the group? First, please send us any relevant studies to update the recent pesticide research report from 2022 to 2025. The original report is the previous 10 years up to 2021. And the Research and Data Gaps Working Group is for scientists and researchers who are concerned with the Bay watershed ecosystem and who are interested in collaborating on ways to educate others about these key issues and concepts. It's an impressive group of scientists, and we'd like to see your expertise added to the team. So please look for an email inviting you to join us at our next virtual meeting in the first quarter of 2025, and reach out to Bonnie Raindrop or me if you're interested in becoming a member. Thank you.
Thank you, Sean. Really looking forward to getting to an annual rhythm with this, where we are bringing to the conference the papers and research findings that we think are really salient, and bringing an annual update, as opposed to the report that we're finalizing, which is a multi-year grab of important research. So throughout the year leading up to the 19th annual conference, we would be glad to receive any tips on important research having to do with pesticides. And we'll be keeping our eyes out for those, and hopefully bring you a summary on an annual basis of important research. All right. Well, thank you to all of our work group leaders and for those updates and reports, and to our participants who have given us input along the way that is helping to shape our work plans. Thank you for that as well. Okay, well, we are up to our last presentation for today, and I'm going to introduce to you Dr. Alexis Temkin. Dr. Temkin is a senior toxicologist at the Environmental Working Group. Her research focuses on pesticide risk assessment and understanding sources of exposure to pesticides and other environmental contaminants. Dr. Temkin began her research career at Columbia University Medical Center, working as a lab technician, studying the molecular mechanisms responsible for environmental influence on gene regulation. As a doctoral student, she studied how exposure to environmental chemicals during development can influence the development of adult obesity and metabolic syndrome. Thank you for joining us, and welcome, Dr. Alexis Temkin.
So good morning, everybody. Thank you so much for having me speak today on the topic of forever pesticides and their forever pieces. And really what this title means is it's a play on the name that PFAS chemicals were given, which were forever chemicals, because of the strength of this carbon and fluorine bond, which makes them very persistent in the environment. And so this talk will be about pesticides that are PFAS, as well as their degradation products, or these forever pieces that might have important environmental implications. And so earlier this year, I was a co-author on a publication on this very topic that was really led by Nathan Donnelly from the Centers for Biological Diversity. And I know he gave a presentation last year at this very conference on the sort of initial findings of this paper. And so I won't spend too much time on this paper, but the idea of this presentation is sort of to give you an update on where this field is going and what we've learned since this publication has come out, since the concept of forever pesticides, or PFAS pesticides, is really rapidly evolving. And so we started this work basically because some publications in Europe were indicating that this process of fluorination of agrochemicals or pesticides was really increasing in recent years, particularly to impart stability in these compounds so that they sort of functioned better as pesticides. And so we decided to take a comprehensive review of what this meant in the United States. And what we found were that pesticide active ingredients were plentiful in terms of those that met the OEC definition for PFAS. About 66 chemicals met that definition, meaning they had a CF3 group that was added to the chemical structure of the pesticide. And those were used in about 24 to 35 million pounds in 2018. One thing we were really concerned about is that These compounds are likely highly persistent, either as parent compounds or degradates, and I'll get into that a little bit later. We also looked at inert ingredients as a source of PFAS, and these are part of the pesticide product. They're just not considered the active ingredient, but they could also meet the definition of PFAS and be a source of PFAS in the environment. And then lastly, we focused on this contamination of pesticide products with PFAS from fluorinated containers. And I know there was a talk at the first part of this conference from a scientist at USGS looking at some of the implications of that as well. And these are just some examples of what those PFAS pesticide looks like. And so really the takeaway here is that this is a diverse group of chemicals with really broad structures, but what they have in common is at least one of these CF3 groups. And some of them are very highly fluorinated, some are used in really high amounts. Some of them have been detected in water, and then some of them all the way on the right here are these inert ingredients. And so these are just some of the chemicals that I'll be focusing on in the rest of this talk. And as I mentioned, the goal of this is really to look at what trends are happening in PFAS active ingredients, how that might differ from country to country, state to state even, so that we can effectively make sort of priority and policy targets based on those trends and how much data we know. I'd also say we're kind of in this information gathering stage. Like I mentioned, Europe has sort of been setting the stage on this research for a little while. And I think here in the US, we're still starting to catch up a bit. So first of all, in terms of the number of approved PFAS pesticides, that number actually differs depending on where you look. So in the US, we have about 66, which makes up about 14% of all of the active ingredients approved in the US. But in the EU and France, that is almost half. There's about 37 approved in the European Union, 30 in France, and then even a fewer amount in Denmark. Interestingly, even though there's fewer approved chemicals in the EU and France, that 12 to 14% seems to still be consistent. And that's the amount of PFAS active ingredients as a percentage of the total pesticides that active ingredients that are approved. And so in the US, this is a figure from our paper. One thing that we saw was that in terms of the percentage of active ingredients that are PFAS, that number was increasing in recent years. So if the pesticide had been approved in the last 10 years, there's, 30% of those were PFAS, whereas on a whole, 14% of them were PFAS. So that might indicate that there's a trend here where pesticides are increasingly becoming fluorinated or meeting this definition of PFAS. Interestingly, in France, there was a report from Pesticide Action Network that saw an increase in sales in these products. And similarly, there was a recent publication from Poulsen et al. This year that looked at the detections of these pesticides in food and also saw that that was increasing. So hopefully I'm going to be able to give you a little bit of info on what their use is like in the United States, and I'll focus on California as well as Maryland, and then what the presence of these are like in the US food supply as well. And so for anybody who researches sort of pesticide use and what that looks like on a state level, everyone turns to California because they have, not only are they a really big agricultural economy, but they have really good data when it comes to pesticide monitoring. So the state's required to report the amount of pesticides used and the location in which they're sprayed. So what I did here was basically take those groups of 66 PFAS active ingredients that I was talking about and look to see how much of those had been applied in California from 2000 to 2022. And so these graphs essentially show the years on the bottom, the pounds applied on the y-axis, and each color represents a different PFAS pesticide. And the graph here on the left represents all of the pesticides applied that are PFAS, of which there were 50 different ones. And the graph on the right here just has a couple of them removed so we could look at some other trends. So if you look at all the PFAS pesticides, one thing to notice is this big orange bar, which seems to be decreasing. This represents an herbicide called trifluralin. And this is actually a trend that you see nationwide as well, where it was sort of heavily used in the 90s, and then its use has been decreasing. And so if you sort of remove that from the graph here, you see that there actually is an increased trend in terms of the amount of pesticides applied. And some of them, oxyfluorophane seems to be the most prevalent, as well as an increase in penthiopirid. And then some of these others are a little bit lesser volume. I should say that this makes up about 2 million pounds of the pesticides applied, which is about 1% of overall pesticides in California. One thing that's kind of interesting about these pesticides is that although they don't seem to make up a large percentage of sort of the pounds applied, because of their persistence, they might be more likely to be detected in the environment. And we saw that with some initial water sampling data that we were looking at that's published in our paper. And so if we look at this data from a more regional perspective, in Maryland, we can look at the pesticide use surveys that are collected from 2014, 2020, and 2022. And I should just mention that these data are a little bit more limited than the ones in California. This is more of a voluntary survey where less than 10% of the farms are actually reporting pesticide use. So this could be potentially an underestimate, but because we're sort of working in a limited data space, having some data on pesticide use is far better than not having any data here. So these graphs are organized the same way. And really the takeaway here is that there are different trends and different pesticides used in Maryland compared to California. And that makes sense, but we still have around 200,000, 150 to 200,000 pounds sort of annually being used of these pesticides in Maryland. One interesting trend here is this decrease in fipronil. One thing I want to mention, though, is that fipronil can also be used in flea and tick medicines, and that can be a significant contribution to fipronil detected in waterways. The other thing I want to mention over here on this graph on the right is this potential increase in cyhalothrin, which is an insecticide. So again, regional and specific differences in how these types of pesticides are used, which could be really important for, potential regulation or policy that might want to be used in the future. And so coming back to potential food detections and that study that I mentioned earlier on an increase in PFAS pesticides as reported in Danish food, what they found was 11 out of the 40, so roughly about 28% of these PFAS active ingredients could be detected in food, and that those were increasing if you looked over time from 2004 to 2022. What I think is interesting here is that there are far fewer PFAS active ingredients approved in Denmark and the EU compared to the US. So if you actually look at this in US foods, what we see is that 33, as opposed to 11, PFAS active ingredients are regularly detected in US foods. And that's about, 50% or so of those PFAS active ingredients that are approved for use in the US. And this data comes from the USDA Pesticide Data Program, which collects thousands of samples of different commodities each year and tests them for hundreds of pesticides. And so I looked at 52 commodities that were sampled from 2017 to 2023 and found that about 39 of those commodities had at least 5% of samples with one or more PFAS pesticide. And really, that's just a way of saying that these are sort of widely detected in US foods. And these are primarily fruits and vegetables that are collected. Some of the more prevalent ones are fludioxonil, bifenthrin, and fluopyram. And I'll show you a little bit more specific data in these next slides. So this is a figure that shows essentially those 39 commodities that had at least one PFAS pesticide detected in 5% of samples. And then this y-axis represents just the number of PFAS pesticides detected. So there's a range here. Some of these fruits and vegetables like watermelon, tangerine, sweet potatoes only have one PFAS pesticide detected, but others like kale, grapes, sweet peppers, have 6 to 8. And these commodities over here on the left, these do tend to have more pesticides detected on them, likely just because of sort of the pest pressures of growing these crops. But that is not unique to just, PFAS pesticides. So they're sort of widely detected across fruits and vegetables that are sort of readily consumed by the US population. And then if you just flip this the other way to say, okay, well, which pesticides, these ones down here on the bottom, are often found. This is a representation of how many commodities they're found on. You see that again, fluopyram, bifenthrin, and fludioxonil are some of the most prevalent ones. And I just color-coded this by fungicide, insecticide, and herbicide. There's just trifluralins, the only herbicide down here, to sort of show you that these are primarily readily detected, but not unique on their function, right? So you'll find fungicides as well as insecticides, not so many herbicides, which would make sense for sort of a food analysis. And what's sort of interesting is, although these are the ones that are most prevalently detected, all 52 of the commodities that I looked at had at least one detection of a PFAS pesticide. So again, widely detected in US foods. And so I want to switch gears a little bit to talk about the breakdown products of this group of pesticides, specifically this metabolite or degradant called TFA, or trifluoroacetic acid. Basically, any pesticide that has this CF3 group, if you remember the images I showed in the beginning, can essentially break down into this ultra short chain PFAS. And we're beginning to learn that this might be sort of a terminal or primary degradation product of a variety of different PFAS sources. And the reason that this is important is because particularly when EPA is registering pesticides, they're required to do short degradation studies to sort of assess what those degradants might be. And you do see that some TFA forms in those studies, but not necessarily in every pesticide product. And even more importantly, the EPA doesn't necessarily consider it that relevant because it only looks at the toxicity of the degradants. And currently, the thinking is that TFA is generally less toxic than some of these parent ingredients, but I would caution that and just say that's sort of a huge data gap that's just beginning to be understood. What's interesting about regulations in Europe is that they tend to look more at the persistence of a degradant as a potential problem. And we know that TFA is going to be extremely persistent in the environment because, again, it is a PFAS and it can't really be degraded any further. And so while we have some understanding that TFA is a terminal degradant, I just wanted to show that this is a very complex process that we're still beginning to understand. This is just the degradation pathway of one of those PFAS pesticides. And so there might be these other intermediates formed. This might vary depending on the environment that the pesticide is used in, but they're all really important considerations for where, when, and how we use these pesticides. There's also been some recent studies that show now that we're starting to monitor for these short chain, ultra short chain PFAS, they're really dominant in these environmental samples. So if you're looking at dust in somebody's home, you might find that these short chain PFAS make up most of the sample that you're collecting. And I think that's also been true in some water samples as well as some human blood samples. And the reason we're beginning to be so concerned about this is that there have been a couple really good review papers, one which was just published, I think, a couple months ago, that's showing that these numbers of TFA in the environment are wildly increasing in a lot of different samples. So this graph just shows all these different places that you might measure or find TFA, like plants and animals, human serum, water, dust. And the comparison here is between the green bar, which are pre-2010 samples, and the red bars, which are post-2010 samples. And in virtually every case, the red bars are higher. So just indicating overall increasing contamination of the environment with TFA. And there's a variety of sources this come from, notably pesticides is on here. And this figure on the bottom here just shows that actually it's very hard to remove from drinking water. So that's another potential concern. And as I mentioned, pesticides are one of the places that TFA can get in the environment, but there's some research that shows they might actually be a really dominant source. So this data comes out of Germany, which has really good water monitoring data for TFA. And what they're finding is that basically in areas of agricultural activity, you have higher TFA in the drinking water or in surface water concentrations compared to, less agricultural areas. And another study that pointed to really TFA coming from agricultural sources was they're present in food. So when you looked at conventional food compared to organic samples in Europe, you also saw that there was higher TFA in those conventional food samples, which I think is the thought is that it primarily comes from TFA contamination in fields where they can be taken up by those fruits and vegetables. It might also be the use of PFAS pesticides on food and the potential degradation of those on the food samples. But here in the US, we actually don't have any TFA sampling in US foods that I'm aware of. And our water sampling is also really scarce. But this group in Europe actually put together a map that showed the estimated TFA potential from the use of all these PFAS active ingredients that I've been talking about. And they basically estimate that there's about 1,500 to 5,000 tons. This is really widespread. A lot of that is coming from just 5 active ingredients, 4 of which are actually not approved in the European Union. And as you can see in the figure, there are lots of regional differences depending on the crops that are grown in these areas and which pesticides are used. And if you look at Maryland specifically, these estimated potential TFA levels from PFAS active ingredients is somewhere around 25,000 to 85,000 pounds of TFA annually. And that can come, ranges in a variety of different counties, primarily ones that have higher agricultural use. And pesticides applied are going to contribute more to this potential TFA formation in the environment, and that it can be a potential groundwater contaminant. And so just to follow up on some takeaways and regulatory recommendations, we made sort of a series of recommendations from our findings in the paper, and I think they still apply here as well with some of this newer data. Basically, that use patterns and trends are likely regional and crop dependent, and that understanding the persistence of these chemicals is incredibly important, and in some cases can really be considered as a basis for regulation. That's something that we see in Europe, particularly because if you're emitting a lot of a persistent contaminant without fully understanding its toxicology, that can have really bad implications in the future. Generally, there needs to be more comprehensive monitoring across the board, increased disclosure of these inert ingredients that I talked about at the beginning. And then I wanted to just touch on this immunotoxicity testing because I saw in some of the questions from the first day, there was a lot of questions about, well, these are approved ingredients. They've gone through toxicity testing, but there are actually huge gaps in our understanding of toxicity of pesticides, particularly because the EPA has started waiving a lot of these immunotoxicity studies. And we know that PFAS that from really well-studied compounds tend to impact the immune system. So again, that's something that we definitely need a lot of data for. Other data gaps are just where these chemicals are, particularly in agricultural communities, as well as where TFA is and where some of these degradation products are. And then understanding sort of the co-exposure to all of these, I think will have really big implications. Again, that was a really great presentation from our USGS scientist who was at the first part of this conference, talked about looking for pesticides as well as PFAS, which could have potential environmental and public health implications. So thank you so much for listening. I am more than happy to take questions, and I'll end here. Thanks a lot.
Okay, Alexis, that was absolutely amazing, incredible work. So we do have about 10 minutes here. And there are some questions appearing. So the usage data that you have shown you've shown California data, and I think you use USDA national data in a few cases as well, plus the Maryland reported data. Is that right? Did we have all 3 of those sources?
Yeah, exactly.
Okay. All right. So yes, that USDA data is useful. And I think we saw in the Maryland data a bit of variability because we're not really sure how powerful that set of data is, but whatever usage information we have can be informative. So just wanted to be clear about that. And California has been collecting that data for quite a while. What do you know when they started with?
I believe they started in 1974, actually. So they have pretty strong records in terms of really precise locations of pesticide use, the amounts applied, which pesticides were used. So it's really valuable data for trends use and for researchers to look at, what might be the impacts on residential communities nearby some of these applications. And it's definitely helped support some of the, I think, policies around, buffer zones and determining when pesticide exposure events might happen. So it's a really important data source as well.
Yeah, yeah, we're jealous in the watershed because we'd love to have data of that quality, but we use USDA and other sources to the best of our ability. Okay, some questions in here about toxicity and our understanding of toxicity. And just wondered if you could elaborate on what kind of criteria are out there that we can use to better understand maybe like residue levels on produce. When does it become a real risk? It actually makes me think of fish consumption advisories. Like, do we need advisories on produce? But what do we know about levels that really create health risks?
Yeah, it's a great question. So I'd say that at least the way pesticides are regulated by the EPA, they sort of set these maximum residue limits for the amounts of pesticides on food. And typically, most food samples are below those sort of legal limits. I think, unfortunately, there is some basically data gaps in understanding of how those are set. For instance, as I mentioned, these PFAS pesticides, many of them don't have immunotoxicity testing because of this sort of process by the EPA to waive this requirement, since they did this analysis that sort of showed it wasn't as useful for their risk assessment process. But if you sort of look at that data through the lens of our understanding about PFAS, which is that a lot of the well-studied PFOS, PFOA actually impact the immune system at really low doses, not knowing how those PFAS pesticides affect the immune system sort of leaves a question in our understanding about how they might actually impact health. So that was sort of a data gap we assessed. And then similarly with the residues in food, there's really limited understanding about how they actually interact together. So, I showed that you might actually find 8 different PFAS pesticides, and that's just part of maybe the 20 different pesticides that might be found on certain samples, how those actually interact together. And there is some epidemiological data that shows maybe higher consumption of high residue fruits and vegetables can have potentially negative health effects. So it's what we're sort of learning to understand, but that immunotoxicity gap, I think, is a big piece that probably should hopefully be addressed.
Okay. The data around TFA is generating a lot of interest, and I guess folks are wondering how quickly does that transformation happen?
Yeah,
Yeah.
that's a great question. I don't think we necessarily know. So in some, as part of the basically degradation studies that are required by registrants to register pesticides at the EPA, they typically, I think, have to do around like 30-day studies. And as I mentioned, in some of those, for instance, there's one pesticide, sulfofenicil, actually showed TFA as a terminal degradation product. So they saw formation of TFA. Sometimes I think for other pesticides, it might take a longer process. There also might be different sort of abiotic factors in the environment that either speed up, slow down that process. But what we have seen, right, is that some of that TFA monitoring, particularly in places where they're looking for it, the levels of TFA are increasing. So it is coming from somewhere, and that just understanding that pesticides might be a source and could be a dominant source in places of high agricultural use. I think just calls for more monitoring to really understand that process. But also some precautionary approaches, knowing that TFA is likely going to be a degradant of a lot of these pesticides simply because of that terminal CF3 or the addition of the CF3 group.
Mhm. Yeah, okay. You showed a map that was, I think, TFA prediction given different agricultural uses, I think. And I noticed, for example, high likelihood in the southeast. So Carolinas to Georgia. Is that because there's a particular crop that's using a
Yeah,
PFAS?
I think it's detailed pretty well in that paper. And so the authors used basically the USGS estimates of county level overall pesticide use and just subset it to the amount of PFAS pesticides that were monitored for. I should say that those data, while they're probably the best estimation that we have for nationwide usage, not all pesticides are monitored, and especially some of the more recently approved pesticides are not going to be necessarily included in that data set. But I think particularly in that region, it comes from really high use of that herbicide trifluralin. I think on a lot of like cotton and sort of high commodity crops, potentially soybeans and things like that as well.
I was going to guess cotton. That red zone there is the cotton zone.
Yeah.
So that may be the story. Okay. Yeah, so an interesting point here that you are showing that increase in TFA over time, through different points in time, TFA increase. We know that one of the challenges with pesticides is the way that formulations change over time, sometimes driven by weed resistance or pests, other things. That change the makeup over time. Are we aware of any big shifts to active ingredients or pesticide formulations? Because we know it gets in other ways in addition to the active ingredient. Any particular product that might explain that trend?
Um, that's a good question. I'm not really sure that we have sort of like the resolution to understand that. I think especially with these with the inert ingredients also too, an important point is that there's really poor disclosure or there's really no disclosure of the inert ingredients in terms of what products they're using. So for instance, in our paper, I think the most frequently used inert ingredient was in 67 different pesticide products, but you actually don't know which ones. So you can't really determine where those are used, what amounts they're used in. So unfortunately, it's a huge transparency issue, I think, and just helping us get a full understanding of the scope of the sort of potential contamination of pesticides with PFAS and then, the subsequent environmental contamination, either with those parent compounds or degradation products.
Hmm. Okay, lots more to learn there. So, Alexis, how about your future research? What are next steps for you, and where is this headed next?
Yeah, I mean, I think primarily a lot of our work is actually understanding what other people are doing and trying to sort of connect those dots. So there's a lot of work in Europe sort of on the TFA front. For instance, there's a, proposal there to sort of classify the toxicity of TFA potentially as having reproductive toxicity. There's some drinking water limits and surface water monitoring limits that are starting to be set there. So I think a lot of our work is really just understanding the scope of that and how things are evolving in different areas and different places.
Okay. Well, your work here is absolutely on point with the interests of our project, and really look forward to whatever is coming next for you, and would love to have you rejoin us in the future because, again, this was just right on target with what we're trying to come together to understand better and find ways to reduce risk through management practices. So right on target there. Thank you so much,
You.
Alexis, and we'll look forward to seeing you again.
Thanks a lot.
All right. Okay, well, thanks to all of our presenters today. Really fabulous set of information. And we're going to use what we've heard from you all to design next year's conference and design work among our work groups. So thank you everyone who has participated in chat and Q&A and given us a lot of information to ponder. I'm going to hand it back over to Bonnie.
Thank you, Greg. Well, excuse me. We wanted to also give you some additional information that might help inform your work with some of the connections that you heard here today. And again, we'll be sending out the roster of everybody who registered, so you'll be able to do some additional networking. And we also wanted to share with you the bit.ly link to the Pesticides and Chesapeake Bay Watershed project page, which is bit.ly/watershed project. And then you can also link to our websites, mdpestnet.org, goorganicnow.org, and saferdisinfectants.org, which really have a real wealth of information and resources and research that we think could really be helpful to you in your work and in your life. So, you know, please copy those down, those URLs down. They'll be in the chat, and visit these websites and give us feedback. Also, in regards to feedback, we could have the next slide. We wanted to just really tell you to look for an email with a survey link that you can give us feedback on today's program and last session as well. And that will come along with your chat and QA transcripts from today. And we also, I think Sean is putting a link to the survey in the chat, so you can take that survey right away. And then you'll see a screen at the end that'll give you another chance to grab that link. So we really take everybody's feedback very much in the process of evaluating what we can do better. And lastly, I really want to thank this year's wonderful conference presenters, Linda Birnbaum, Emily Woodward, Greg Allen, Vicki Blazer, Anna Rule, Alexis Temkin, Veronica Corella, Cleo Braver, and Sean Lynch. Everybody did such a great job. We so appreciate what you did, your work, and bringing it to us this year. And we also want to thank our conference team, our moderator, Greg Allen, Emily Ranson at the webinar controls, Sean Lynch moving a lot of parts behind the scenes, and Doug Miller for video editing, and he also wrote the original music scores for the conference. And to all of you for joining us today. We'll be emailing you with the chat and QA transcripts, again, the stakeholder community contact information to help you connect with colleagues, and video links to both sessions, and working group invitations. We'll also send you the finished products that you heard about from our work group leaders over the coming months. And we're very grateful to this year's funders and sponsors for their support of the Pesticides and Bay Watershed Project. And since it's Giving Tuesday, if you'd like to help support this project, we've put a link in the chat that you can use to make a tax-deductible donation to Maryland Pesticide Education Network. As a small nonprofit with a very large and growing agenda, we're very appreciative of your support. Thank you all for all that you're doing to protect and enhance the Chesapeake Bay watershed and our ecosystem population. Here's to a really fruitful 2025 for all of us. Thank you. Have a good rest of your day.
And thank you, Bonnie. You are the glue that holds this project together. So thank you very much for that. And to everyone, we will see you at the 19th annual conference. Thanks, everybody.
Bye, everybody.